In this lesson, we're going to take a look at applied and environmental microbiology. Applied microbiology is the commercial use of microorganisms. These microorganisms can be used for a number of different things, and we really divide applied microbiology into three different categories. Food microbiology is the first one. This is going to be the use of microorganisms in food production. Also in food microbiology, this is including preventing food spoilage and then also food-related illnesses. Industrial microbiology is the use of microorganisms in industrial manufacturing processes and looking at solutions to environmental, health, and agricultural problems. The third category is going to be environmental microbiology. This is exploration of where microorganisms are found in nature, and it's also the study of how microbe activities affect other organisms and also affect the environment itself. We're going to start off talking about food microbiology more specifically. With food microbiology, there are a number of different effects that microbes can have on our food. The first of those is going to be flavor. They do contribute to the overall flavor of our food because they produce certain molecules that are going to be found then in our food. They contribute certain aromas to our food. They also affect the consistency of the product. They can be used for food preservation and then also for nutritional improvement of that food product. The last one, not something beneficial at all. Microbes can contribute to food spoilage, and that is a real problem that we do have to worry about when we are talking about food production and then also food storage. Looking at food production, microbes are used a lot in food production, and this has gone on for many, many years, centuries. Saccharomyces cerevisiae is a yeast, and this particular yeast metabolizes sugars, and the result of that metabolism of sugars is going to be that it leavens bread. And that leavening means that it's going to raise the bread, and we see an example of leavened bread or raised bread in the image right here. The way that that is going to happen is because the metabolic actions of the yeast itself are going to result in the production of carbon dioxide as a product. That carbon dioxide is a gas, and that gas production is going to create the holes that you do see in bread products. If we're talking about something like sourdough bread has bacteria that help to contribute to its flavor. And in that case, the bacteria is generally added using a starter culture of some sort, and that bacteria is going to produce lactic acid. And that lactic acid is going to contribute that characteristic sour flavor to the bread. In general, when we're talking about food microbiology, the use of the word fermentation refers to any desirable characteristics or desirable changes that occur in the product due to microbe growth. This is in contrast to spoilage, because when we use the term spoilage, that's going to be any undesirable or negative impacts that microbes have on our food products. There are a lot of different fermented foods that result from microbes being present. Fermented vegetables, typically these are going to result from lactic acid bacteria. Here we can see a list of 4 different lactic acid bacteria: Streptococcus, Leuconostoc, we have Lactobacillus, and then we also have Lactococcus. Different types of fermented vegetables that we can have: kimchi and sauerkraut. These are both going to be fermented cabbage. Soy sauce is fermented soybeans and wheat. Chocolate is actually a fermented product. This is going to be produced by fermented cacao seeds. Coffee, there's also some fermentation that goes into its production. The fermented coffee berry is going to release the coffee bean, and then the coffee beans are roasted after that. And then another one would be pickles. When we're talking about pickles, there are different ways to produce pickles, but the ones we're talking about here are going to be pickles that use microbial fermentation as the acid source that's used in their overall preservation. The last one here is an animal feed product. So we don't just have human consumption products on our list here for fermented vegetables. Silage is something that is used for animal feed. This is fermentation of potatoes, corn, grass, grain stalks, a variety of different components that are fermented, and then this product is fed to animals. Fermented meat products are going to be produced when we combine fermentation with drying or smoking. In this case, the meat is going to be mixed with a starter culture, and then the meat and the starter culture are going to be incubated at a relatively low temperature, and the fermentation process is going to be allowed to take place. The meats that we're talking about would include things like salami, and then we also have other products like fish sauce, which are really the result of this overall fermentation process. In that case, it's fermentation of fish that would be producing the fish sauce. Fermented dairy products are going to rely on the action of lactic acid bacteria, just like the fermented vegetables. In the image right here, you can see examples of fermented dairy. We have buttermilk, yogurt, a variety of different cheeses that are produced this way. And if we look at how this product is going to be produced, it does typically rely on starter cultures being added. And if you look at the diagram here, we're going to start with a dairy product like milk, and that is going to be pasteurized, which will kill any unwanted microorganisms that happen to be there. So that's going to be a lot of the microorganisms that would affect the flavor of that actual milk product. Then we're going to take the pasteurized product, and there is going to be a starter culture that will be added. And this is typically going to be a starter culture that contains some of those lactic acid bacteria. We're then going to allow that fermentation process to take place. This is going to produce some curds, and then those curds are going to be filtered out and collected. That will result in a lot of the different cheeses that we do have. And besides the ones right here, we could also have things like cottage cheese, which is not shown on this particular slide. Alcohol fermentation is using microbes for the process of converting simple sugars into ethanol, which is really drinking alcohol, and then also carbon dioxide. So there's really two products that are being produced in this case, and we have different types of alcohol fermentation products. Wine and spirits is one group of them, and if we look at the basic process of that, we're going to start with generally some type of fruit material, and there's going to be some preparation involved where that fruit is going to be cleaned up. It's also going to be crushed. So here we can see grapes, and we're looking specifically at wine production here. And then those grapes are going to have a starter culture added to them. That starter culture is probably going to contain yeast and then also some bacteria. They're going to be added. They're going to be allowed to actually ferment. And as they ferment, they're going to produce that carbon dioxide and also that alcohol. And then we're going to clarify the wine basically by some type of filtration process. Once we've done that, the wine is going to be placed into barrels. It's going to be aged. During that time, the yeast will continue a little bit of their fermentation, but we're also going to get some flavors from the actual barrel that it's being contained inside of. And then the last step is going to be bottling of the product. That's the production of wine. If we're talking about the production of spirits, the spirits are going to have an additional step where we're going to have some distillation, which is going to help to concentrate that final product. Beer and sake are also fermentation products that are taking advantage of the alcohol fermentation process, but they are going to be produced by a slightly different process. With these, we're typically starting with some type of grain material that is going to provide our food supply for the yeast or microorganisms. In this picture here, it's barley that we're starting with. And one of the first steps that's going to occur is that we're going to take the barley, which contains a lot of starch, an example polysaccharide, and we want to break down that starch material so that we can have sugars that are more easily metabolized by the yeast. So we have a couple of different steps where we're going to try to get those starch molecules converted into sugar. This involves allowing the grain to germinate some. It involves mashing it up and mixing it with some water so that we get even more enzymatic activity. And then we're going to kill off ultimately the microorganisms that are there. Once we've done that, we're going to add the yeast culture. The yeast culture that is then added is going to metabolize whatever sugars are present, and then we're going to essentially get our beer production when the yeast produces that ethanol and then also the carbon dioxide. And then that can be aged, it can be filtered, and it can be pasteurized and bottled up to get our finished product, which is the beer or the sake. Vinegar is also a food microbiology product. This is produced when the ethanol from fermentation is metabolized a second time. So that ethanol that we get is going to be oxidized to form ultimately acetic acid. There are oftentimes going to be specific starter cultures that are added to perform this conversion. We do have a lot of different vinegars that can be produced, and this is really depending on what the initial sugar source is, so whether it's apple juice or some other products, and then it also depends on the starter culture that's being used, and that's going to give us different flavors in the vinegar that is produced. The other side of food microbiology that's not food production is going to be food spoilage. Food spoilage is going to involve adverse changes in nutritional value, taste, and then also the appearance of the food. So these are all obviously negative things. Food spoilage can be the result of intrinsic factors, and it can also be the result of extrinsic factors. So if we talk about what these are, intrinsic factors will be factors that are inherent properties of the food product itself. When we're talking about extrinsic, this is going to be factors that are due to other things. So factors that are due to maybe the way that the food was handled, maybe the way that it was processed ultimately. But both of these are things that can result in the food spoilage down the road. If we talk more about the intrinsic factors in food spoilage, first of all, nutritional composition is going to play a big role. This will determine what types of microbes are present and whether those microbes are going to grow or not. Basically, what we're talking about here is what kind of nutritional value can this product provide to that microbe. So if they're very, very sugary, that's probably going to contribute to breakdown by microbes and ultimately spoilage of that product. Water activity is another one. This is really looking at whether there's available water for microbe metabolism. If there's a lot of sugars and other molecules present, then that's going to decrease the available water. So we're really talking about available water, and that is necessary for the metabolism of most organisms. The acidity plays a big role. Low pH values typically reduce microbe growth. So if you have something that has a lower pH, it's more acidic, typically we're going to get less spoilage. And then another one is just the overall physical structure of the product. This might include things like the type of rind or the type of covering. If you think about a thick covering around an orange, that is going to reduce the spoilage. The shell around an egg would be another example. So really this is determining whether or not the microbes are going to be able to access the moist interior of the food. So the moist interior of the egg, the moist interior of the apple or the orange, and products such as that. The likelihood of food to spoil is going to be used to group it into 3 different categories. Perishable foods are going to be the most susceptible to spoilage. These perishable foods, they need to be kept cold. If they're not kept cold, they're going to spoil very quickly. And we're talking about them spoiling in a matter of days if they're not kept cold. So those are the most susceptible. Semi-perishable foods, these are going to be kind of a medium range of perishability. These can be stored in some type of sealed container for months if they're not opened. So this would include things like tomato sauces that we might have, things in a jar. But once we do open them up, they will spoil within weeks after opening. And typically we do have to refrigerate them at that point when we do open that container. The third category is non-perishable foods. Non-perishable foods can be stored pretty much indefinitely without spoiling, and this typically includes dry, fermented, preserved, or nutritionally poor products. And many of these are things like pasta. Pasta can be stored for very long term. Dried beans can be stored very long term. Other examples would be things like crackers and potato chips, things of that nature. A lot of those things that we think of as going stale. To prevent food spoilage, there are a number of steps that can be taken. So one of them is just paying attention to the food processing methods. One common way to process foods commercially is going to be with the canning process. Canning itself is going to use very high temperatures, and it's also going to use high pressure so that most microorganisms that are there, they are going to ultimately be killed. Another way to preserve food or prevent food spoilage is going to be through the process of pasteurization. Pasteurization is not going to be as rigorous as the canning process, so it's not going to heat to as high of temperatures, and it's certainly not going to heat as long, but it is typically going to kill most organisms that would be causing changes in the overall flavoring of the products. Desiccation and drying can be used to preserve foods. This is something that's sometimes done with different meats. Lyophilization would be freeze drying. And then there's different types of radiation that can be used to sterilize or to get rid of a lot of the bacteria and microbes that might be there. Gamma radiation is one. This is a type of ionizing radiation. And then a second type of radiation would be non-ionizing radiation. UV light is a good example of that. And that also is going to result in killing some of the microbes that would be there present on that particular food product. The use of preservatives can also be very helpful in preventing spoilage. Sugar and salt, both of them are going to be able to preserve products using the process of osmosis. So they limit the amount of available water, and if we do decrease the amount of available water, then that is going to really limit or inhibit microbe growth. Jellies, jams, they are using sugar to help preserve them and limit microbe growth. A lot of times different meats can be salted as well, and that's going to help preserve them. Here we have an example of fish. Products like bacon would be another example where salt is being used to preserve. The temperature is very important during the processing and storage process. It can affect the overall food spoilage. As a general rule, higher temperatures are best when we're doing the processing of the food. And then on the other side of it, if we're talking about storage is going to really be best with low temperatures, because the lower the temperature, the lower the metabolic activity is going to be. So that's lower metabolic activity of any microbes that might be present. Food microbiology is going to include the study of foodborne illnesses. This includes both food infections and also food intoxications. Food infections are going to be caused by consumption of live microorganisms. Food intoxications, on the other hand, these are going to be caused by consuming microbial toxins. There is a long list of different microbes that can cause different foodborne illnesses in humans, and this table is really just listing a short list of those. Just to point out a couple of them, E. coli does cause some foodborne illnesses. This can be found in meat and raw milk. We also have salmonella, often in undercooked eggs, some meat, some dairy products. Listeria is also one that is kind of relatively common in dairy products. And one of the things about Listeria is that it tends to grow relatively well at low temperatures, which means that it does oftentimes grow even when we're storing our food at proper temperatures. So that's a look at food microbiology. If we move on and we talk about industrial microbiology, industrial microbiology is going to be the use of microorganisms for producing valuable compounds and as environmental sensors, and then also in the genetic modification of plants and animals. In industrial fermentation, this is going to be large-scale growth of particular microbes for producing beneficial compounds. The process involved generally falls into two categories. We have what's called batch production and then also continuous flow production. Now, with both of these, we have to provide some type of food source or food supply nutrients to the organisms. Now, in batch production, the organisms are going to ferment whatever that substrate that's provided happens to be, and we're going to just wait until that food supply, that substrate, is completely gone, completely exhausted. At that point, we're going to harvest our product. With continuous flow production, it is continuous. So what this means is that we will keep replenishing the substrate for the microbes, and at the same time we have to continually remove the waste. So this is something that's going to be much more involved. It's going to require more complex equipment and more complex monitoring, and we can continuously harvest some of the product from that production. There are various enzymes and industrial products that can be obtained from microbes. Just a list of some of those we can see right here. Amylase is a spot remover. That is an enzyme. Pectinase is going to be an enzyme that can be used to release cellulose fibers from flax, and then that flax can be used for making linen. Protease is oftentimes used as a meat tenderizer. Streptokinase can be used in the medical field because it can dissolve blood clots. We have food additives. Food additives are things that improve color, they improve taste. And then we also have food supplements that can be obtained from microbes. And this includes things like vitamins and also amino acids. In addition to enzymes and industrial products, we also have alternative fuels. Alternative fuels could include things like ethanol, methane, and then also hydrocarbons. Here is a quick look at some of the different pharmaceuticals that can be obtained from microbes. There are a number of different drugs that come from a variety of bacteria and other microbes. E. coli is a common one on this list. You can see that we can get Taxol, we can get human insulin produced in E. coli as well. Some different vaccines can be produced in yeast. So again, just a list of some of the different pharmaceuticals that we can get produced by microbes. Here is just a quick list of some of the products that we've already talked about and the different categories that they fall into. We can have a number of enzymes produced by microbes, food additives and supplements. We can have industrial products such as indigo and then also plastics, alternative fuels, pharmaceuticals, and then also pesticides and various agricultural products.